In this video, we're going to go from gene to a portion of that gene's DNA sequence to the bonds and atoms that are associated with that portion of the DNA sequence. As an example, we're going to use the PRNP gene. It's a prion protein encoding gene, and it's on chromosome 20. There's chromosome 20. Let's expand into the PRNP region. There it is, the PRNP gene. It has two exons. This exon on the right has a lot of SNPs that 23andMe records. Let's expand on that so you can see the letters associated with the DNA sequence. You're going to notice that the sequence has an orientation to it. The top band, ACGT, from left to right is 5' on the left, 3' on the right. Let's actually copy that sequence onto the clipboard. So go ahead and copy the sequence, place it on a clipboard. Let's paste that into a text editor so that we can verify that indeed we've got the GAC GT sequence. There it is. Let's paste those letters just as they are into MarvinSketch, and so we can see the bonds and atoms. All I did was paste that same 5-letter code into MarvinSketch, expand on those. Now you can see that the G is the 5' hydroxy end, and the T on the other end is the 3' hydroxy end. Each of these letters are going to be linked by a phosphodiester linkage. We can expand them all out. It gets a little crowded because there's not enough space here. But there you can see the complete 5-letter sequence, all bonds and atoms associated with each of those nucleotides. The 5' is on the left, the 3' is on the right. Each of those hydroxy groups, we can make them negatively charged, so they're reflecting the pH in physiological conditions. And there we have the complete 5-letter sequence from gene to bonds and atoms.